food, drugs, or ideas, what you consume influences who you become. On the Mind and Matter podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a PhD in neuroscience where I focused on neuroendocrinology and the neurobiology of behavior, and before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to help parse and translate the information that guests share on the podcast. In addition to the podcast, I write long-form written content inspired by the show where I integrate what I've learned across episodes. I also have a free weekly newsletter where I provide you with upcoming guests, share links, and provide commentary on scientific studies and research that I'm reading, and more. Visit mindandmatter.substack.com to find all of my content. You are about to listen to the full episode, but there will be 1 to 2 short ads immediately following this before the conversation. If you would like to hear ad-free episodes and get early access before they release more widely, become a paid subscriber at the Mind and Matter Substack at mindandmatter.substack.com. Becoming a paid subscriber is a way to directly support my efforts, allowing me to make more episodes and higher quality episodes, and also helping to free up my time so that I can spend more of it on doing research and synthesizing results from the podcast and from the literature so that I can do more things like writing on the Substack, putting more content into the newsletter, and getting these things out more frequently. So if you want to support the show and get ad-free episodes, go to mindandmatter.substack.com. Hey everyone, a great way to support my efforts is to visit my support page at mindandmatter.substack.com. A link to that page is in the episode description, or you can search for it on the Substack page itself. The support page has an up-to-date list of my affiliate partners. These are companies I work with, and if you buy those products through those links or using the discount codes provided, those will get you a great deal on a variety of products related to optimizing physical, metabolic, or mental health and vitality. These are all products I use myself, and some of them are directly related to podcast episodes or formulated by Mind and Matter guests. For example, there's a great product called Keto Citra, formulated by a kidney biologist who is on episode number 186. It contains the ketone body BHB together with several minerals in a precise ratio formulated with kidney health in mind. There are also links to products like physical technology devices to track your metabolic health, digital applications to help you find and learn about food and consumer products, and more. One product I use every day is the AquaTrue water filter. I recommend this to everyone because it gets everything out of your drinking water: heavy metals, microbes, endocrine disruptors, microplastics, you name it. Other charcoal or gravity-based filters that a lot of people use don't get everything out, they just get some of the stuff out. So I really like the AquaTrue product. You can look for a link to my affiliate partners on my support page or directly in the episode description to this episode, or you can go to mindandmatter.substack.com to find it there. All right, Dr. Greg Steinberg. All right, Dr. Greg Steinberg, thank you for joining me.
My pleasure, Nick.
Do you want to start off by just telling everyone a little bit about who you are and what you study?
Yes, of course. So my name is Greg Steinberg. I'm a professor in the Department of Medicine at McMaster University and a co-director of the Center for Metabolism, Obesity, and Diabetes Research. Our lab has really been focused over the last 25 years at understanding cellular energy sensors and how they respond to changes in nutrient availability and energetic stresses.
And, we're going to dig into a lot of detail and nuts and bolts in this conversation with one of these pathways called the AMP kinase pathway. I want to give you a chance up front just to maybe kind of step back. And so like you're like a cell biology and a biochemistry guy. We're going to get into some of the nuts and bolts and mechanisms and a lot of detail about this important pathway in cells. But if we just sort of step back and take a bird's eye view, can you just talk a little bit about how you think about like what cells are doing generally in terms of bioenergetics and metabolism? Like you can pick a cell type if you want to, or tissue system, but like what are sort of the basic high level ways or some principles that cells are using to sense what's in the environment and figure out how to funnel and transform and utilize energy?
That's a big question to get started. Not so easy, but certainly, cells are interested in maximizing efficiency, and whether that means using the available resources for the maintenance of cell function, maintaining ATP production, really depends on the kind of nutrients that are available at any given time. And the hormonal cues, especially that have adapted to link fuel selection with energy demand. And so these cells are always, just like people, allocating their checkbook per se,
Yeah.
what they want to do with their resources. Do they want to invest in the future, or do they want to use those resources for the here and now, right? And so every cell is making that decision every second of the day, depending on numerous levels of inputs, whether that be, as I mentioned, hormones or the number and type of calories that are coming into the cell.
Mm-hmm. So cells basically, I mean, I like the analogy that you use. They have to decide how to use the resources. We want to use them to build stuff now, to repair and maintain, to invest in the future. So, at a high level, a cell is going to have to always sense what's coming in, how much energetic potential is coming in, what kind of substrates do we have, glucose, fats, all of those things. What's coming in? What do we have? Is everything working? Are things broken? Do we need to fix things? And then somehow, there's like a time component here. A cell must have some sense of like where it is in the cell cycle, how long it might live or how long it might have to live, and thinking about things, like you said, like an investment in the future. And so there must be mechanisms in cells that enable it to play around in all those dimensions.
Yeah, absolutely. The mechanisms are very complex and multifaceted, and even spread over not only time, where there's acute versus chronic renovations per se that must be done in the cell, that are more acute issues versus down the track, but also energy dependent, depending on the type of fuel that's available. Is it carbohydrates right now? If we have a lot of carbohydrate available, then it makes more sense to do this with this fuel. If we have a lot of fat, we're fasting. Well, we can use this fuel very efficiently to do this job in the cell. And, one of the things that we're very interested in is how the cell not only responds to this energetic stress, which is sort of a cell signal, such as when our adenine nucleotides drop, which are the AMP and the money currency per se, but also how it responds to these changes in nutrient availability, such as the fats and carbohydrates. And we really think that the AMP activated protein kinase is a central gauge that sort of integrates all these inputs together, along with these hormonal cues, to partition out these resources as the cell sees fit.
Mm-hmm. So there's this thing called AMP kinase that we're about to talk about. And the thing that people are probably mostly familiar with that'll be listening is ATP. This is the cellular energy currency of a cell, adenosine triphosphate. So there's ATP and then there's ADP and AMP, and we're talking about AMP kinase. So can you kind of break this down for us? What are these things? What is the M versus the D versus the TP? And then what is AMP kinase?
Yeah, so, as you mentioned, touched on there, these are the energy currency. So if we think about money, these are the actual things that we can spend in the cell. This is what ultimately, this is ultimately why everyone dies. You run out of this currency, right? It doesn't matter what you die of. This is why you die. You don't have enough of the high energy currency, which is ATP, and you have a lot of this low energy currency, AMP. We know, as the name suggests, that the AMP activated protein kinase gets switched on when the high energy currency gets all degraded to this low energy state. And, ultimately, this is what causes cells to die and organisms to die, is when we cannot maintain this high energy currency state.
Yeah, so it's in a sense, it is like you run out of money. You have to keep track of how low the bank account is. And I imagine just like in our own lives, If your bank account starts to get too low, some alarm bells go off. Hopefully, you've got some checkpoints and some systems that can detect that. So it sounds like the cell is going to really care about how much ATP versus AMP there is. And I would imagine AMP kinase is probably one of potentially many mechanisms or places in the cell that's listening to this AMP ATP ratio, which is going to be a clear and important signal about how much energy might be available to the cell.
Yeah, absolutely. So the key goal of the kinase here is to get switched on before that high energy currency, that ATP, is depleted, right? And so it gets switched on when we get increases in the intermediate molecule, ADP, and AMP start to rise. And, small changes in these energy currencies, it detects it very, very quickly. And what it does in this scenario is that it switches off things that are not essential for the survival at this time, right? And switches on processes to make more of this, make more money per se for the cell so that it can survive. And so, if you think about this in the a housing analogy, for instance, your roof's going to break down in 10 years. But your toilet's overflowing. You want to fix the toilet probably before you fix the roof in right away. And so it directs resources to where it needs to do this to do immediate needs. And,
Got it. Yeah. So if we stay with like a housing analogy, everyone's probably had these experiences at some level. If you're starting to run out of money and things are really tight and we're in a recession, you're going to shut off luxuries that you don't absolutely need. You might not go on vacation, but you need to pay your mortgage, you need to pay rent, you need to buy groceries. Sometimes you're in a boom phase, things are going well. Now you can splurge, you can get the bigger TV, you can go on vacation, whatever it may be. So AMP kinase in this analogy, it's part of the detection mechanism for how much of this energetic currency the cell has, how much ATP versus ADP, and it's going to sense that and help direct the resource allocation within the cell, help it spend smartly depending on how limited resources actually are.
yeah, exactly. It's going to decide what is the priority for that cell to, get over this stress. Is it transient? Is it just a acute sort of issue due to increased demand? Or is it going to be a chronic stress that needs, bigger inputs and bigger investments to survive?
Yeah. And I think we're going to talk quite a bit about sort of nutrient sensing and functional effects and the allocation of resources, how these things are sort of smartly happening within the cell. But first, I just want to give people a clear picture on like what this machine is and where it is in the cell. So if we just start with sort of the basic enzyme mechanics here. So AMP kinase is an enzyme. Just what is this protein? What is an enzyme? And what is this particular enzyme? And what is its fundamental enzymatic function? What is it doing?
Yeah, so it's a three-part protein, so it's a heterotrimer. It has a catalytic subunit. It's a kinase, right? So fundamentally what kinases do is that they get switched on through a phosphorylation event. This phosphorylation through the catalytic subunit then phosphorylates other things in the cell, which means it either switches on these other things or switches them off, right? And so this is transferring of a phosphate group, which then affects the activity of other enzymes in the cell, which then influence all sorts of different metabolic pathways. And, over the last 25 years, AMP kinase, AMP activated protein kinase, has been shown to phosphorylate over 100 distinct substrates across the cell. And so this happens in the cytosol, it happens in the mitochondria, it happens in the nucleus. And so there's distinct pools of AMP kinase, which can control a lot of different biological functions of the cell. And it really integrates these nutrient and energetic stresses through this cascade.
Yeah. And so, I mean, phosphorylation and dephosphorylation, this is a really fundamental aspect of cell biology, right? Like there's constantly molecules and proteins being phosphorylated, phosphate groups added, or dephosphorylated coming off. If we go back to AMP, ADP, and ATP, this is really just adenosine mono, di, or triphosphate. So you have 1, 2, or 3 phosphates. So these phosphate groups, we could almost think of that as the energetic potential of a molecule, right? The more phosphate groups there, the more of those bonds that exist on the molecule, the more energetic potential there is. So that's why ATP is the high energy version of AMP.
Yeah, absolutely. And the kinase, we have the catalytic subunit, and then we have two regulatory subunits. As I said, it's a three-part enzyme. And these other regulatory subunits bind to these adenine currencies, adenine nucleotides, and can sense very precisely if there's changes in this ratio between the high energy currency and the low energy currency. And this is fundamentally what gave the kinase its name. 25 years ago plus, actually 35 years ago plus, when when it was observed that the kinase, there was a common kinase which inhibited both fat and cholesterol synthesis. And people thought that these were two different kinases until Graham Hardy and Dave Carling really identified that it was a common kinase that controlled both pathways and was sensitive to these changes in adenine nucleotides.
Mm-hmm, mm-hmm. And so I think one of the things we'll probably keep coming back to is this is what you might call an integrator or a master regulator enzyme. It's not just phosphorylating one type of molecule. It's doing lots of different things. It's not just listening to one type of nutrient, like just glucose or something like that. It's listening for lots of different signals and integrating those signals together. So AMP kinase, a kinase is something that phosphorylates, so it can add phosphate groups to something. And the something in this case is many things. AMP kinase can phosphorylate many different things in the cell, not just AMP, it sounds like. Where in the cell is this located? You sort of said, I think, that there's different pools of AMP kinase. I imagine it's not located in just one place, but does it tend to concentrate? Is it just sort of floating through the cytosol and it's kind of homogeneously spread throughout the cell? Or does it localize and concentrate in certain places like the mitochondria or the cell membrane or some other place?
Yeah, we really don't have a full understanding of how this subcellular localization is occurring. And originally it was thought that the kinase was primarily in the cytosol, but more and more we know that there's specific substrate targeting motifs within the kinase, specifically the beta subunit, targets it to potentially lipid rafts, it targets it to nuclear abundance, and so and it targets to mitochondria, and it depends on the kind of stress that it's ensuing, depending on where it goes, so that it is localized with the kind of substrates or the things that it needs to phosphorylate and control. So depending on the inputs, the kinase will move. It might be in the cytosol, but if it's a mitochondrial stress, or there's a lipid stress, it might move to the mitochondria to phosphorylate proteins, which will increase fat burning, for instance. So we're really at the infancy as far as our understanding about exactly how the kinase is moving. And, this has been one of the challenges because there's 12 different combinations of the kinase. You have 3 different subunits. You have multiple isoforms of each of these subunits, and we're just really starting to understand about the subcellular localization and how the kinase is moving, given the expanding technology advances which are making this possible.
Got it. So there's complexity and room for regulation and some interesting dynamics happening at multiple sort of scales here. So this thing's regulated in time and space. AMP kinase, the cell can put AMP kinase in particular locations in the cell. So it's not just like a membrane protein where like this is a receptor that's always going to be embedded in the plasma membrane, and that's just kind of, it gets made and then it goes there. This could be in the cytosol. It could localize to the mitochondria. It's going to have different temporal dynamics, I imagine, based on ATP, AMP levels, and all of these things. And there's different subunits. So it's like a 3-part, multi-part kinase, a multi-part enzyme, and then there's different versions of each of those parts. So like roughly speaking, it's sort of like, I've got a camera set up here, but I could swap one lens in and one lens out. I can use a different battery hookup. It's the same machine, but I can sort of use different versions of it depending on my needs.
Yeah, this has been a real challenge. There's 12 different combinations. And so we're just really figuring out maybe this combination is the one that does this. maybe this one is the one that responds more acutely to when we exercise or when there's a mitochondrial stress. Maybe this one gets switched on when there's a lot of fat around. And, this is
Right.
one of these intricacies that we really want to start to understand. And, we really think that AMPK isn't just a fuel gauge, it's sort of an energy asset allocation manager per se, or, it's controlling these metabolic traffic jams that are occurring throughout the cell. And there's precise pools of AMPK which do this. Some might be more sensitive to these changes in adenine nucleotides, but what we're learning more and more about is that it's more than just detecting, whether the lights are on or off. It's or whether there's, energy coming out of the mitochondria. It's actually affecting the flux of nutrients. And we really think this is a much more nuanced view of the kinase, not just making a cell survive from a near death event, but much more important actually in, daily fluctuations as we go from fasting to refeeding. It, fine tunes the cell's utilization of these nutrients.
Yeah.
And this is sort of a new view, which we've really expanded in recent years by understanding how the kinase responds to different nutrients.
Yeah, yeah. So, I imagine this is becoming, more complicated and nuanced over time. I imagine it's, you sort of, I think, just said it's not merely a fuel gauge. It's not merely is energy high or low. There's a lot more to it than that. It's like what type of energy is present, how do we need to use it, all of these things. But it's sort of like the highest, most basic level of understanding. What does AMP kinase activity look like, or when is it high versus low? Is when the cell's running out of energy and you're in more of a fasting or a starvation condition, is AMP kinase activity going to crank up really high, and vice versa? How do we just start to think about it at like sort of the highest, most coarse grained level possible?
Yeah, I mean, AMP kinase is super high. You can think of, altitude training, low oxygen levels, fasted. These are conditions when there's mitochondrial stress, low oxygen. The cell is really trying to survive. Hypoxic conditions. These are the kind of signals that are really going to activate the kinase. When is the kinase low? Well, if we eat a lot of food directly after a meal, whether that be high fructose, high glucose, lots and lots of sugar, oxygen sitting around, the kinase is really, really low, right? So the fuel gauge is super full, the kinase is low. When the fuel gauge is low or high, as in after a meal and we're just relaxing, the fuel gauge is full and, the kinase is really, really low. And
Mm-hmm.
so these are sort of the two extremes, obviously, in this scenario. And unfortunately, we spend more and more time in the full fuel gauge these days than the low fuel gauge. And, as a result, the kinase is suppressed from doing a lot of its normal activities or cell maintenance jobs that it's supposed to be doing.
So on average, for the average person today living at this time, the AMP kinase pathway, the AMP kinase enzyme is not as active as it was on average historically for most humans.
Absolutely, and we think this is a huge problem and a contributor to chronic metabolic disease in that, we are not switching on these cell stress pathways like AMP kinase like we used to in the past, whether it be through inactivity or whether periods of nutrient scarcity. Even people typically don't even go 8 hours without eating, right, anymore.
Mm-hmm.
So it's, that means the kinase is always, on the full side of the equation, on the fuel gauge. And as a result, the cell doesn't undergo the maintenance checks and the kinase isn't phosphorylating the things that it used to.
Right,
Keep the
right.
cell optimizing its function.
Got it. And again, roughly speaking, if we kind of go back to the analogy that we were playing with before, if we think about our own lives, if you get a big promotion and you've got bonuses coming in and like it's a time of abundance, that's when people tend to get a little bit loose and a little bit lazy with maintenance checks, and they don't worry about things so much. But when money's tight, when you have to live very diligently and frugally, that's when you start to get much more precise about, like, okay, I better check to see what's working. I better make sure everything is working properly and we're not cutting corners. That's kind of what AMP kinase sounds like here.
Yeah, absolutely. You could say the cell gets a bit complacent with
Yeah.
doing the daily jobs of restoring mitochondria, whether that be through things like mitophagy, restoring, fixing the things that need fixing, because it thinks it's always going to have nutrients available. It doesn't need to
Yeah.
worry about this, you know. And it's a very unique scenario that really hasn't been around for many years, right?
Right, right. It was rare for this
From
to
our
always be
evolution.
right in evolutionary time, naturalistic conditions. AMP kinase would go up and down regularly. But now, for a lot of people, it's sort of always low because we always have an abundance of energy coming in. So the cell is getting a little complacent. It's maybe not doing all the housekeeping it would otherwise be doing. So it used to be that people, some animals, including humans, had to deal with the raw, cold, hard environment. Sometimes we were starving, sometimes there were famines. But even in today's conditions, it's becoming more and more popular for people to do things like fasting, intermittent fasting, not eating for 1 or 2 or 3 or sometimes even more days as like a health thing. And these are the conditions. Not having nutrients coming in all the time in super abundant levels. These are the conditions that are going to make AMP kinase levels go up. So let's talk about like the fasted condition a little bit. Whether we're forced to fast by nature or we choose to fast today for health reasons, say, AMP kinase activity, this enzyme is going to go up. What are some of the major things that are now happening? When AMP kinase goes up because we're in the fasted state, what's getting switched on? What is the cell starting to do?
Yeah, that's something that's been really fascinating for us to unpack. This idea that, and really led us to think more about what the kinase is doing and how it's regulated, right? Because with fasting, our cells are not at risk of dying per se, with an 8-hour fast, 10-hour fast. And we really got thinking, our energetic stress, our energy currency, these adenine nucleotides in the cell are not changing that much with that fasting condition, which yet AMP kinase was being switched on under these conditions. And, that really led us to think that there must be a metabolite that goes up with fasting that might activate the kinase and elicit these fuel shifts. And, we discovered that one of the key metabolite that activates the kinase with fasting is fatty acyl CoAs. So palmitate, oleate released from adipocytes. So, when we fast, the first signal is that we get changes in hormones, insulin drops, glucagon goes up, epinephrine goes up. This triggers the release of free fatty acids from adipocytes. And this release of free fatty acids activates the kinase, okay? And causes the kinase, AMP kinase activity to be increased. And this is independent of these adenine nucleotide changes.
Yeah, yeah.
And this in turn tells the cell to start burning more fat, start making more mitochondria, start doing more mitophagy, shut down things like mTOR. Because we don't need these growth pathways. And, this is really important adaptation that occurs.
Right, right. So basically, fasting, some of what you said there was like, adipocytes start releasing fatty acids. So literally, like fat is being broken down, fatty acid levels therefore rise. AMP kinase is detecting this, and it's sort of saying, "Oh, we're starting to tap into our energy stores. We don't have a lot of new energy and food stuff coming in. Therefore, don't do things like build new structures." That's kind of what mTOR means.
Yeah, exactly. Let's maximize this fuel that's coming in. This is telling we don't know whether this is going to last 2 hours or 20 days. We better start making plans to become more efficient, right? And to utilize the fuel that we have coming in as best as possible. And so this is really what the kinase is doing when we go into fasting like periods, or we expose ourselves to, say, a ketogenic diet, which is, high in fatty acids. We're really telling our body, it's not all about the calories. It's about, how we best use these available nutrients in the most efficient way possible.
Got it. So again, if we kind of play with this analogy, we keep coming back to, maybe you've got your household, you're running your house, you're taking care of the family, you've got a job, but like the recession is setting in, you're not getting a promotion this year, but you haven't lost your job. So you're going to be like, okay, we have to use the energy coming in well and smartly. We're not going to build an addition to the home. Maybe we'll skip on the vacation. We want to make sure that we're doing the things that are necessary with what we have. We're going to make sure we've got enough to pay the mortgage and the bills. And we're probably not going to build a lot of extra stuff and get a lot of new gadgets in the home because we don't know the next time we're going to be in a state of abundance. We're used to living like this today in history as humans, but our cells are sort of, they've got this whole evolutionary history baked into their DNA, so to speak, literally at some level, and they don't know when the next wave of abundance is going to come. And so there's this sort of economical tendency built in, and AMP kinase is part of that.
Yeah, absolutely. I mean, in that kind of scenario, you might start looking for another job or something, right? Oh, maybe I better do a little work. And that, that's what AMP kinase is doing. It's like saying, maybe we better make a few more mitochondria. We don't know how long this is going to last. We got to make sure that we have a way to make more of this energy currency down the track, right? If times get really tough, we want a lot of extra mitochondria around to make sure we survive and adapt to this. And certainly that's what the kinase does. It improves the fitness of the mitochondria and really redirects those resources towards these kind of adaptations.
Mm-hmm, mm-hmm. And so this AMP kinase, adenosine monophosphate kinase, it's adding phosphates to AMP and to other things. So it's allocating energy in terms of the cell biology. What I want to talk about the nutrient sensing capabilities and dig into this a little bit. So how exactly, we'll talk about the different nutrients it senses and how like different ratios of fats and carbs can affect how the energy is allocated when you have it more in the form of carbs or in the form of fats and things like this. But how does it just sense nutrients in general? How is it detecting glucose versus fats? Like, how does that actually happen? Do these things come in and touch this kinase in a particular location? How does it sense these things?
Yeah, I mean, so we have this three-prong kinase, the catalytic isoform, and we have these two structural structural subunits, the beta and the gamma subunit. And one of the things we discovered is that the fatty acyl CoA molecule, which is the activated fatty acid inside the cell, binds to the beta isoform of the kinase, altering its structure, making it, allosterically active, per se, increasing the ability of the kinase to phosphorylate other things. And also what happens is that it protects it from dephosphorylation. So any kinase has a kinase that puts on that phosphate group as you described, but at the same time it has a kinase, a phosphatase, which sucks off the phosphate off the catalytic subunit. And so it's the ratio between those two things that changes, right, when an active kinase becomes activated. So this conformational change when fatty acid binds to the kinase protects it from being dephosphorylated. It allows the kinase to be in a more active state once fatty acid CoAs bind to it.
Got it. And in general, for people who are unfamiliar with biochemistry stuff, when we talk about phosphorylation, we should think of this as a switch, basically. Like when something is phosphorylated, like an enzyme, it generally means it's switched on and it's going to be more active. But then if you take that phosphate away, you can switch it off. So the moving around of these phosphate groups, phosphorylation and dephosphorylation, this is a way for cells to regulate, what machines and tools are switched on and which ones are switched off.
Yeah, that's right. So the phosphorylation state is more like an on and off switch, while I think about the allosteric control is more like a dimmer, right? Where you can have variations in the activity of the enzyme by altering its conformation shape. And so you really, can activate, change the kinase. So because it can go from, in the fat situation, the fasting situation, you don't need your kinase at 100 like you're at hypoxia or
Yeah,
altitude
yeah.
training and exercising, you only needed it at 5 or 10, not 100, right? And so, this is sort of like we want the lights turned up, but we don't want full on sun shining
Right,
down on
right.
me. And so, this is a real nuanced view of how kinases can be regulated by substrates
Got it,
such
got
as
it.
fatty
So
acids.
yeah, so I've got a light switch in the hallway. It can be on or it can be off. But when it's on, there's a dimmer function to it. So in the evening, I turn it down because I don't want the lights to be super bright. Right now, it's turned all the way up because it's the daytime and I need to see stuff. So literally at the molecular level, there are structural features on proteins and enzymes like AMP kinase that allow them to be switched on or off, but also sort of more finely tuned in terms of how on they are and how much juice they're using.
Yeah, exactly. So in addition to this, subcellular localization we discussed, as well as, how the cell it goes from location to location depending on the time, we also have this dimmer switch to control how much activity is there. And this then depends, dictates how what substrates it might phosphorylate because every protein will have a different affinity for being phosphorylated. Some proteins we know, for instance, one protein, acetyl-CoA carboxylase, a key protein controlling fat burning, is acutely sensitive to AMP kinase. This substrate recognition motif is very, very optimally designed for AMP kinase, whereas other proteins, potentially things related to mitochondrial remodeling, might be less sensitive to small changes in AMP kinase. And so, this also provides us with a more nuanced view that, we can control AMP kinase, but then there's also sensitivity of how AMPK can phosphorylate other things down in the cell.
Mm-hmm.
We don't want all 100 substrates phosphorylated every time AMP kinase gets switched on.
Right, right. Yeah, you want to be efficient, right? Because that all of that takes time, it takes energy, and you don't want to do it haphazardly.
Exactly. For instance, something like autophagy, every time the cell gets a little stressed or fuel utilization changes, we don't want to be killing everything or packaging everything out and throwing it in the garbage right away, right? We want to save that for maybe when energetic stress gets a bit worse.
Yeah, yeah. So if we imagine the average person on what we might call the standard American diet. So they're eating more than enough calories. They're probably eating more calories than they need. So they're going to be in more of an energy storage state. They're going to tend to gain weight. They're eating fat and carbs, so an abundance of carbohydrate and fat substrates. They're going to be in they're not going to be in autophagy mode. They're not going to be in a fasted state. They're in the opposite. So AMP kinase is going to be very low. And so the things that AMP kinase turns on will not be on so much. They'll be off because AMP kinase activity is low. They're not going to be doing a lot of this cell maintenance stuff. They're not going to be doing as much mitophagy and recycling of things and all the stuff that we touched on. Let's say that such a person then decides, you know what? I want to change my lifestyle a bit. I'm going to start fasting. Walk us through what then starts to happen in their cells with respect to AMP kinase.
Yeah, I mean, even with an acute fast, going 8, 10 hours, once we deplete glycogen levels within the liver, we're really dependent on uh, fasting, and in particularly lipolysis, increases in free fatty acids to maintain sugar production coming from the liver,
Mm-hmm.
which is energetically costly. And, switching our cells to conserving glucose for the brain. So we switch all our cells to utilizing fat. And, we think that this is where AMP kinase becomes quite important. It's this fuel partitioning, not necessarily the total energy that's really, really critical here. It changes the fuel source, not necessarily the calories that's really driving these adaptations. And this is where it can start happening fairly quickly with a fasting kind of paradigm.
Yeah, yeah. So even when we say fasting, we might just mean a few hours of fasting, 6, 8 hours, something like that. Maybe someone starts there, they're doing like intermittent fasting, and they're not fasting for days at a time, they're fasting for hours at a time. That's it sounds like in that approximate time frame, that's going to be enough to start to run down glycogen stores and things like that. And so the cell will start to partition its energy differently. It will start to get more choosy about which organ is getting the glucose versus the fatty acids. Is that basically what we're saying here?
Yeah, absolutely. It will start prioritizing burning fat and saving the glucose, right? And that's designed to save that glucose for the brain because, our glucose levels, as I tell the endocrinology students and yeah, fourth year biochemistry students, the amount of sugar in our blood is just so small, right? It's like 2 tablespoons of sugar floating around, and our body
Mm,
maintains it within that tight range. We can eat Diet Cokes this big, or we can not eat for days, yet our blood glucose stays level, right? It's just remarkable how we can maintain that, that kind of fidelity around the glucose control. And that's done because we shift the way our cells use the fuel.
got
And
it.
AMP kinase, and we show that, AMP kinase is really central to that.
Yeah, yeah. And when someone is, in their standard American diet mode and they're eating what is now today a normal amount, so they're eating all the time, they're never even fasting really. Is AMP kinase low or does it get shut nearly off? Does it like how low is low here?
Yeah, no, AMP kinase is really low in that setting, undetectable in most cell types. And, this is why one of the reasons why we need to get up and move more, right? Exercise elicits a metabolic stress, not just on the muscles, but our heart, all our tissues. And, this sort of prompts the AMP kinase to become activated. And this is one of the initial things that really got people excited about AMP kinase as a potential, exercise signal. And it holds up as a key exercise signal that gets switched on as soon as we start moving into moderate levels of activity.
Yeah, yeah. So AMP kinase very low. It can be basically undetectable in most cells if people are fully sedentary, fully eating the standard American diet. That type of thing. Exercise or fasting will start to turn it on. When someone goes from, say, mild fasting, not eating for a few hours, to now saying like, okay, I'm going to try and do like a 2 or 3 day fast where I'm eating very little for 2 to 3 days. AMP kinase is going to go up even higher. What happens in that case compared to just fasting, say, for a few hours? Do whole new activities in the cell start to get switched on, or is it just like a quantitative difference? You're just going to get more of the same things coming on for longer.
Yeah, what will happen in that kind of setting is that AMP kinase will phosphorylate transcriptional programs
Hmm.
related to fasting and mitochondrial programs to increase more mitochondria, increase fasting related programs in the cell. So we have the acute changes, increasing fat burning at the level of the mitochondria, but then we're talking about making more mitochondria or destroying, eliciting autophagy and other programs that could be, more long-term solutions. And this is sort of how the trajectory goes.
Yeah,
Each
yeah,
requiring, more and more energy. Obviously, transcriptional programs and switching those on and off is costly, right? You don't want to start making a whole bunch of genes and a lot of protein if you're immediately going to shut it down, right?
yeah, yeah.
So this is the more nuanced view where longer fasting will turn on these transcriptional programs.
Yeah. So if you fast for longer, to put it more abstractly, if a bigger change happens that requires a more drastic shift in a cell, those shifts often amount to new gene programs getting turned on or off. And to tie this back to some of the fundamental biochemistry stuff we were talking about before, the way that happens in terms of nuts and bolts is something like AMP kinase might go up we're fasting for 2 days, AMP kinase goes up. It's a kinase that phosphorylates things. One of the things that phosphorylates might be transcription factors, proteins that go into the nucleus and then touch the DNA and start turning on new gene programs. So it sort of all comes back to this energetic patterning within the cell. Something like a kinase, like AMP kinase, will go up. Transcription factors and other things get phosphorylated. They get switched on. They can now start turning on new genes, which basically means the cell can behave differently. It can do things it couldn't do before, or conversely, it might shut down things it could do before. But it's all about passing these phosphates and these sort of switches back and forth to different proteins to dictate what's happening based on what's kind of coming into the cell and what it has to work with from an energy standpoint.
Yeah, absolutely. I mean, I think about this more, I'm an endurance athlete, right? I did triathlons, long distance triathlons, swimming, these kind of things. When you let yourself get out of shape, then you start training, the first thing you notice after you come back from a long bike ride or something is that you're just craving carbs, right? You're just hungry for carbs. But after you've been training for 6 weeks, the biggest change is that you've trained your body to use fats more efficiently. You don't crave carbs the same way because you've trained your muscles to burn fat a lot more efficiently. They've adapted. They've made more mitochondria. And the same thing is happening over this fasting duration. The same thing is this exercise training paradigm where, people who are chronically endurance trained are much more efficient at burning fatty acids and don't require glucose to the same degree to keep their cells happy.
Interesting. And so I want to talk a little bit more about like mitochondrial dynamics. So, and there's multiple ways to think about this, right? Just the total number of mitochondria in a cell. Do you have more? Do you have less? Do you have a lot? Do you have little? There's also sort of like the size and shape of these things. And there's a lot of detail we could get into. But at a high level, so if we go back and we think about someone eating a standard American diet again, they're sedentary, an overabundance of energy, AMP kinase is low. They start to fast and exercise more. They start to eat less. They're maybe explicitly fasting and or exercising more. So from an AMP kinase standpoint, they're cranking it up. AMP kinase is going up. What is starting to happen at the level of the mitochondria? Is AMP kinase in general going to increase the number of mitochondria? And if so, how does that actually happen? How does that actually sort of get instigated within the cell?
Yeah, so AMP kinase will do really three distinct things to mitochondria. One, it will start off by if you're inactive, you probably have a few mitochondria that have been sitting around there for a long time, right? These are not optimally efficient mitochondria. So they're not running like a well-tuned furnace in your cell, right? Your furnace hasn't been tuned up, your car hasn't been tuned up for years. And this is what your mitochondria are like. So one of the first things it's going to do is do mitophagy to get rid of these, poorly functioning mitochondria. And AMP kinase is really critical for getting rid of those inefficient mitochondria as a first step.
Got it. And so the mitochondria, these are the structures that are just transforming energy for the cell. They're making all of the ATP or most of it. And they can sit there idle just like we can as a full organism, or just like some other device we might use can. I remember being a kid and during the dead of winter, we had a diesel truck, and dad would turn on the diesel truck, but then he would let it sit and run for a minute because you didn't want to just start driving it right away. You got to let it sort of warm up. We can roughly think of the mitochondria in this fashion, like depending on how much or how little they've been used, they can kind of get out of tune or need some warming up to work efficiently.
Yeah, absolutely. They have a half-life. They need to tune up. You need to change your spark plugs in your car every few thousand miles. And, if you aren't using your car, these things get gummed up. And that's really what the mitochondria are. And so, that's one of the things that AMPK will do. It will also, so in addition to getting rid of these sort of inefficient mitochondria, it'll turn on programs to make more mitochondria at the same time, right? Mm-hmm. So we're going to get rid of these old, dysfunctional, not optimized mitochondria, and we're going to start making new ones. And this is sort of the mitochondrial biogenesis side of the equation that is replacing these old damaged, non-optimal mitochondria with brand new mitochondria. And this takes a bit more energy, right? And but over the long term, it's definitely worth it.
Got it. So in other words, if AMP kinase is always low, if someone is always sedentary, they always have an overabundance of food, they're never fasting, they're never exercising. One of the reasons this is not good is because the AMP kinase pathway, the AMP kinase enzyme will always be very low. It will never go up, and therefore the cell won't be swapping in new spark plugs and making new mitochondria, and it won't be cleaning up and recycling some of the machinery that enables it to actually effectively utilize and use that energy.
Yeah, absolutely. That's one of the first things that goes, and you want to replace these spark plugs, get in new ones there. And then the third thing that happens really, we have the mitophagy, we have the mitochondrial biogenesis. The third thing is that, which can happen much more acutely, right? This happens before anything actually, is that the AMP kinase phosphorylates proteins on the mitochondria, which affect their shape. And this is called mitochondrial fission or fusion. And this then allows the fuel selection to be optimized, right? And so you get pinching of the mitochondria and changes in the mitochondria, which can happen within minutes or hours of fasting, really, where you're getting these adaptations so that these mitochondria, even if they're not optimal, they'll start burning more fuel more efficiently.
Got it. Got it. So, I mean, mitochondria really are like smaller cells within our cells. They can grow, they can divide, they can be killed and replaced, just like the whole cell can. So that's what fission and fusion are. So basically, AMP kinase in a state of nutrient abundance, high energy, we're not moving around a lot. AMP kinase is low. We start moving, we start fasting, these things. AMP kinase goes up. Among other things, it's actually going to phosphorylate or add these, on switches to proteins on the mitochondria. This will cause them to fuse together or break apart. This might cause them to grow. This might cause them, I would imagine, to be targeted for recycling. I imagine some of them get just completely broken down, recycled, and then we just build whole new ones basically from scratch.
Yeah, exactly. That's that's the process here. The ones that aren't up to snuff, get thrown in the recycling bin through that mitophagy process if they don't respond well to that initial stretching and fission and fusion process.
Yeah. So, and that's how things work when we're relatively healthy. AMP kinase is sensitive to the environment. It can detect all of the things it needs to detect. It can do all of these things. If it's working, I would imagine that in various disease states, say the AMP kinase itself or things closely related to it can start to break. And then that can be a huge problem because AMP kinase is this sort of master integrator regulator thing that is doing this sort of very important fundamental energy allocation functions within the cell. I'll let you sort of pick the disease state or whatever you want to start with here, but what does it start to look like when AMP kinase itself is compromised somehow?
Yeah, so the most one of the most obvious settings where we know AMP kinase becomes dysfunctional is in obesity and insulin resistance, where the kinase has an abundance of substrate or fuel all the time, and it's chronically switched down. And as a result, we're not managing our sugar levels appropriately, right? We're not switching. One of the hallmarks of insulin resistance or diabetes is the inability to switch between fuels. So what you have is a lack of metabolic flexibility where when you eat a high sugar meal, you don't switch to burning that sugar. And as a result, the sugar builds up in the blood and we develop type 2 diabetes. So we're not able to adapt to these shifts in substrate availability from fasting to refeeding like we should. And so this is one of the most obvious examples where AMP kinase dysregulation occurs. And, this is really where how we first got interested in AMP kinase by the understanding that when it gets switched on in muscle, for instance, it causes the muscle to start soaking up the sugar from the bloodstream. And AMP kinase is central to increasing glucose supply to that working muscle to fuel it. And as a result, blood sugar levels start to decline in people with diabetes when we exercise. And, if we develop drugs to AMP kinase that directly target the kinase, we see the same thing, right? Blood sugar levels decline, and we get better and more healthy mitochondria in muscle.
And so so one of the things too I want to ask you about is there's lots of ways for nutrients to be present and abundant. They can sort of all be abundant. In some sense, that's what the standard American diet is. Like there's plenty of fat around and plenty of carbohydrate, and we're eating protein. Like all of the things are present. There's also ways for things to be relatively abundant, but sort of skewed in one direction or the other. You might be eating a high carbohydrate diet. You might be eating, say, a ketogenic diet, which is becoming more popular. So there's enough energy around. There's a lot of inputs coming in, but they're all fat and not very much carbohydrate. What are we learning about how AMP kinase sort of detects and responds selectively to carbohydrates versus fats of different kinds? There might be multiple ways to talk about this, and I'll let you kind of choose what's best. But just given the increasing popularity in the ketogenic diet and ketosis, what sort of happens in that state when someone has enough energy, but it's a ketogenic diet, it's all fat, low carb? How is the AMP kinase detecting and responding to that differently than it might, say, a high carb or a standard American diet where you've got fats and carbs present?
Yeah, so this is something that's really interesting to us. I mean, it's not about, and it builds on, I guess, the message that it's not just about energetic stress, right? These adenine nucleotides that regulate the kinase, because we know what ketogenesis is doing is it's really mimicking the fasting condition, right? We get an abundance of free fatty acid, these fatty acyl-CoAs. These will activate the kinase and cause this fuel partitioning to cells to use more fat. So, it sustains sort of the fat metabolic traffic, right? It pushes everything to make more mitochondria, more fat, and fat burning. And so it's really mimicking the fasting-like condition. And this brings us to one of our favorite hormones, actually, which is, if we think about AMP kinase being the intracellular fuel gauge, it brings us to one of our favorite molecules, which is the molecule that coordinates metabolic stress with the rest of the body, being the growth differentiating factor 15, GDF15, which is secreted from cells and tells the brain to stop making increasing food abundance. And we've shown that this hormone gets switched on with the AMP kinase stress pathways, and that then it communicates with the rest of the body to tell you to eat less fat. So it's a feed forward system to communicate fat load. And this is one of the reasons why some people lose weight on a ketogenic diet.
So in the ketogenic state, if you're doing a ketogenic diet, on the one hand, you're not fasting, you're not eating, there's food coming in, you have energy, but at the same time, at a cellular level, at a biochemistry level, you are mimicking many of the aspects of fasting. So you're simultaneously eating something, but your cell is sort of under conditions that are similar to fasting in certain ways.
Yeah, I mean, I think it's important to note that 95% of the people who say they're eating the ketogenic diet probably aren't. It's quite a degree of very limited carbohydrate consumption, right? Which is super hard to achieve under today's dietary conditions. Right?
Yeah.
I mean, I think that's an important caveat. But when you get to a true ketogenic diet, very much you're eliciting the same metabolic programs as fasting, and that's really without actually while still consuming calories.
Yeah. So if you're eating the standard American diet, AMP kinase is cranked like all the way down pretty much. If you're totally fasting, not eating, especially for extended periods, you're going to crank AMP kinase up. If you're doing a true ketogenic diet, you are in ketosis, but you're eating as much as you want. So you're eating fats, very, very little carbs. You're in ketosis. Where is AMP kinase in terms of its overall activity levels? Is it closer to being on the low side or the high side, given that on the one hand, there's energy coming in and nutrients being sensed, but on the other hand, you're in this fasting-like state?
Yeah, I mean it's going to be cell dependent, but I mean our view is that you're sort of in the mid-range fuel tank, right?
Got it.
It's sort of the kinase is being tickled, it's being turned on, it's telling the cells to make fat burning, and certainly that's what we saw in mice. If we adapt them to a super high calorie, high fat meal. What starts happening if we make a mouse with a single mutation where these fatty acid CoAs bind, they don't get the signal to start making more mitochondria. They don't get the signal to start burning fat to the same degree. Will they still respond to the adding nucleotides and the stress? Sure. But, we're sort of in the mid-range game where you're starting to, make changes to the way the cell allocates resources to become more efficient with the ketogenic diet.
Got it. So if you do eat a ketogenic diet, there is evidence to suggest that essentially the cell will get better at burning through fat because that's sort of what it has to do.
100%.
Yeah. Yeah.
Yeah.
Interesting.
And, we think AMP kinase is really central to that adaptation.
Yeah. I want to talk a little bit about the relationship between AMP kinase, this pathway that we've been discussing, and mTOR. mTOR is another sort of master regulator in cells. It's another nutrient sensor and the sort of integrator of many signals. And people think of mTOR as like the main sort of anabolic pathway. When mTOR is high, nutrients are present. You've got a lot of materials to work with. You can build new structures. So if we're building muscle or we're getting bigger, and mTOR is going to be high. So if we start to think about like the sort of the matrix of possibilities here, fed versus fasted, mTOR high or low, AMP kinase high or low. The standard American diet, the average person today, relatively sedentary, not exercising that much, eating a lot, carbohydrates and fats. We already said AMP kinase will be very low under these conditions, and I would imagine mTOR would be relatively high under these conditions. And is the way to think about this that when AMP kinase is low and mTOR is high, the body's basically going to build more tissue and get bigger?
Yeah, absolutely. I mean, there's sort of the yin and yang of the body. They're intimately connected, cellular energy sensors, right? And AMPK is the low energy gauge. And when the energy gauge is full, mTOR gets activated. And, these things are connected biochemically through phosphorylation events, right? AMP kinase phosphorylates and inhibits key subunits of mTOR, which inhibit its activity. So AMP kinase is actually upstream of mTOR in many cases, which is consistent with its ability to respond to a variety of internal cues, whether it be nutrients as well as adding nucleotides. So it shuts down mTOR. So you definitely do not want to be building things.
It makes sense.
Under energetic stress. And one of the easiest ways to shut down those programs is by inhibiting mTOR. And that's certainly what AMP kinase does.
Got it. So if you are under energetic constraints, you're fasting or whatever, AMP kinase goes from low to high, and it's going to start phosphorylating stuff and doing its kinase activity. Among other things, one of the things it ends up doing is inhibiting mTOR, which should make logical sense to people, right? If energy is limited, you don't want to be building up new structures that weren't there before. You want to be doing some of these other things, recycling and swapping out the old mitochondria, autophagy, all of that stuff, maintaining and recycling and being efficient with the energy that you have, which is limited. It's only when energy is super abundant and you don't focus on all of those things that you then want to say, switch on mTOR and start building brand new structures.
Exactly. Yeah. So that's right. And, there's more and more connections being understood, actually, that mTOR can also fine tune AMP kinase activity. So, not only does AMPK potentially inhibit mTOR, there's inputs that maybe mTOR is acutely sensitive to, maybe certain amino acids like leucine, that then could fine-tune AMP kinase activity. So there's a real feedback loop between mTOR and AMP kinase to balance, the allocation of resources.
Yeah. Does AMP kinase, is it sensitive to particular carbohydrates and particular fatty acids? I would imagine it is at some level. Like, can it tell the difference between glucose and fructose on the carb side, and can it tell the difference between like saturated and unsaturated fats on the fat side?
Yeah, we don't know about that much about that. We know, fructose compared to glucose, when it's metabolized, can have quite distinct effects on ATP levels. And in that way, fructose versus glucose can have distinct effects. Our studies have really not delineated much of a difference between saturated and unsaturated fatty acids with respect to the AMP kinase, though.
Yeah. I want to ask you about AMP kinase and cell metabolism stuff in the context of cancer as well. It's becoming increasingly appreciated that, my take on sort of the whole realm here is that, most people, most of the time, decades ago and years ago, tended to think of cancer as mostly a genetic thing. It's going to be all about getting mutations in the right genes to get a cancer cell to form. Today, it's becoming more common for people to see this as more of a metabolic thing, at least some of the time. That when the cell becomes metabolically dysregulated, then cancer can form. It may not be all about, or even mostly about, mutations in DNA. When we think about the other thing that people are talking about too, that I want to mention up front is, fasting, autophagy, that stuff. It seems to be that when people fast intermittently, when you're doing the autophagy and things, one of the things that often gets cleaned up are these sort of precancerous cells. And like, there's a benefit there to fasting sometimes from a cancer prevention standpoint. And I'll let you comment on that where you see fit. But when we think about cancer and the formation of cancerous cells in relationship to AMP kinase and, states of energy abundance versus the opposite, how do we start to think about this, and what are we learning there? How does the AMP kinase pathway tie into, say, anti-tumor signaling versus pro-cancer events in the cell?
Yeah, I mean, like many things, it's a complex answer. Whether it's a pro-tumor or anti-tumor. As we talked about, it's really the last-ditch survival mechanism. So if you're trying to kill a tumor cell, AMP kinase with a chemotherapy or radiation, AMP kinase may help that cancer cell survive. However,
Mm.
from a cancer prevention aspect, um, what we know, and this is really what put a lot of pieces together with respect to AMP kinase research, is that we know that the tumor suppressor protein, LKB1, is an upstream kinase that activates AMP kinase. And so LKB1 is a common mutation in many cancers, and it activates AMP kinase, suggesting that, mutations in AMPK activation are associated with more cancer.
Hmm.
In addition, there was a large body of epidemiological evidence, initially in the early 2000s, linking the AMP kinase activator metformin. Metformin activates AMP kinase by, acting as a brake on the mitochondria. This increases AMP kinase activity, and there was a large body of epidemiology to suggest that potentially metformin may have anti-cancer effects. And certainly, people without diabetes, that might not be true, but there was certainly a lot of evidence to support that from a cancer prevention standpoint, activating AMP kinase, inhibiting mTOR could be very beneficial for cancer prevention.
Mm-hmm. Yeah, so the effects of AMP kinase being high or low could depend on whether the cancer cell is already formed or not.
Yeah, absolutely. It depends where they are in their stage of progression. Have they gone to a metastatic stage where they're dependent on different fuels, oxygen conditions, lots of different areas. But I think preventing cancer, it's quite clear that AMPK, really elicits a lot of positive effects to prevent tumor development.
Yeah. And I'm not sure we mentioned this explicitly, but is AMP kinase present in all cells? I would imagine it's pretty much in all cells, but even if that's the case, is there a lot of heterogeneity in terms of organ and tissue systems and how much AMP kinase they tend to have?
Yeah, this is every cell has some AMP kinase, whether you're a mammal or right down to plants, actually, there's AMPK homologs. And so in yeast, and so every cell has it. But as I mentioned, there's all these different combinations, 12 different heterotrimeric combinations, which uh, make it challenging, and certain different heterotrimers might have mutations or be higher expression in tumor cells versus the other tissues. So this can influence the activity of the kinase in cancer.
Yeah, yeah. One of the sort of high level things that I'm detecting in this conversation, but also other conversations I've had about similar things, mTOR pathway, different aspects of metabolism. This AMP kinase pathway, just like these other pathways, wasn't under naturalistic conditions for most of evolutionary history. These things weren't really built to always be on or always be off, to always be high or always be low. They're sort of meant to wax and wane and have this sort of yin yang relationship. We talked about mTOR and AMP kinase. They're sort of meant to wax and wane in different ways. And if you're always in the high state versus the low state, that's probably not good because, as we mentioned, when you're always in the low AMP kinase state, the sedentary, infinite fuel abundance and nutrient abundance state, AMP kinase is going to stay low. And then you're not going to be, swapping out the old spark plugs and recycling your mitochondria and all that stuff we talked about. Is that, just in the abstract, is that how you think about it? That, these pathways are meant to be in the sort of yin yang up down relationship. They're not always supposed to be on or off, but that's sort of what modern society has, jammed them into.
Yeah, absolutely. As we, started the conversation talking about through evolution, we went through periods of fasting and refeeding, which are largely absent these days. And certainly the AMP kinase would have a lot of diurnal variation and, week to month variation, depending on fuel sources and even seasonality. Where I am right now here in the Toronto area, it's minus 15 and snowing, right? We can imagine that under these conditions, our lipolysis would be activated if we're trying to survive in these conditions. We're really changing, the fuels we're using and the metabolic stress our bodies are under. And so this is a really important aspect, and I think something that's really challenged the field from a drug discovery standpoint. Ever since the discovery that exercise and metformin activate AMP kinase, people have been trying to design ways to make drugs to more directly target the AMP kinase to elicit whether it be beneficial effects in type 2 diabetes or weight loss or of exercise and metformin to potentially exert beneficial effects. And, one of the challenges the initial drugs had is that they were more like a sledgehammer towards the kinase, where, they tried to design the best drug possible that switched on the kinase 100% of the time at 100-fold, right? Bright lights blasting at it continuously. And as you might anticipate, there were problems with that approach. It's just like, acute going for an exercise bout, going for a 45-minute run is okay once a day, but running for 45 days straight, 24 hours a day is not going to go too well for you, right? Your body's going to break down. And certainly that's what they found. The biggest problem with these drugs is that they cause your heart to get much bigger and cardiac hypertrophy because they were turning on the kinase in all the cells all the time. And so there were complications associated with that, consistent with, going out running continuously 24 hours a day, which probably is not a good idea for anyone.
Yeah, yeah. What are some of the active areas of research in your lab or in the field generally to do with AMP kinase that you think are particularly exciting and that are going to start to tell us some really exciting new things in the next year or two, say?
Yeah, so there's been a huge, uh, when I started working on AMP kinase, there were probably just 10 or 15 papers a year on the AMP kinase, and now there's 20 or 30 papers a day coming out, right? And so the field has certainly expanded, but there's still a lot to understand, specifically about how the AMP kinase responds to different nutrients. Changes in glucose. If fats are going up, there's things that are changing simultaneously. How does the kinase decide which out of all the different things it can phosphorylate around the cell, how does it decide where to go and when to do it? Right? We're just really beginning to understand that. So there's many more nuances around key substrates which connect fat burning, mitophagy, fat oxidation, and glucose oxidation, and this fuel switching that really are incompletely understood. And I think using, time resolved and spatially resolved technologies will really help us get better answers around this.
Yeah, yeah. But I think one of the sort of practical high level takeaways I'm getting from you with respect to AMP kinase is, AMP kinase is this critical sensor of nutrient abundance. It's a critical regulator of lots of stuff in the cell. And the problem the majority of people have today is that AMP kinase is too low too often. We live in a state of abundance and we live these sedentary lifestyles. One of the reasons that's bad for health and it's connected to chronic illness is because the AMP kinase pathway within the cells is always low. And one of the consequences of always having low AMP kinase is you don't have a lot of this recycling and maintenance happening that would swap out old mitochondria for new mitochondria that would stimulate autophagy and do these things. So if AMP kinase is always low for whatever reason, things are in your cells will basically fall into a state of disrepair and just not work as efficiently.
Yeah, absolutely. And, one of the things we're learning more and more about is things like metformin, which act like a brake on the mitochondria, they activate the kinase, and we see some beneficial effects. But, we are also know that things like Ozempic, GLP-1 will also activate AMP kinase, increase AMP kinase in different tissues. And in fact, in the brain, lowers AMP kinase, which leads to reduced food intake. And so it's quite interesting that this could be an important part of the food response to GLP-1 like
Interesting.
drugs.
Are the GLP-1 drugs having a direct effect on AMP kinase, or is it an indirect effect because appetite is going down and people are eating less?
Yeah, I mean, what we think is happening, although there's no direct evidence, the GLP-1 receptor is really highly enriched potentially in the pancreas as well as the brainstem. So it might be having direct effects on these tissues to lower AMP kinase, and there's a biochemical pathway to potentially explain that, where the G-coupled receptor, the GLP-1 receptor, is phosphorylating, leading to phosphorylation of AMPK, which inhibits its activity, inhibitory phosphorylation. But the subsequent restoration of AMP kinase in peripheral tissues is probably secondary to the weight loss and improvements in glucose homeostasis, in our view.
Are there any final thoughts you want to leave people with about AMP kinase and cell metabolism generally, or anything you want to reiterate from our discussion today?
I think, we had a great discussion. I think it's a exciting area to continue to explore how nutrients interact with the kinase, and hopefully we'll continue to learn more about how our bodies adapt to different physiological stresses and the importance of AMPK in that area.
All right. Well, Dr. Greg Steinberg, thank you very much for your time.
Yeah, thanks so much, Nick.
Hey everyone, a great great way to support my efforts is to visit my support page at mindandmatter.substack.com. A link to that page is in the episode description, or you can search for it on the Substack page itself. The support page has an up-to-date list of my affiliate partners. These are companies I work with, and if you buy those products through those links or using the discount codes provided, those will get you a great deal on a variety of products related to optimizing physical, metabolic, or mental health and vitality. These are all products I use myself, and some of them are directly related to podcast episodes or formulated by Mind and Matter guests. For example, there's a great product called Keto Citra, formulated by a kidney biologist who is on episode number 186. It contains the ketone body BHB together with several minerals in a precise ratio, formulated with kidney health in mind. There are also links to products like physical technology devices to track your metabolic health, digital applications to help you find and learn about food and consumer products, and more. One product I use every day is the AquaTrue water filter. I recommend this to everyone because it gets everything out of your drinking water: heavy metals, microbes, endocrine disruptors, microplastics, you name it. Other charcoal or gravity-based filters that a lot of people use don't get everything out, they just get some of the stuff out. So I really like the AquaTrue product. You can look for a link to my affiliate partners on my support page or directly in the episode description to this episode, or you can go to mindandmatter.substack.com to find it there.